feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, hello, and welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity Webinar. Thanks for joining us. This is my first show of the year, so I'll add Happy New Year to that. I was in the States a lot over the holidays and came back a few days ago, and I realized that Singapore does have winter. If you weren't here, it got all the way down to 22 degrees. So I guess the world's changing. We have a really special episode tonight featuring a fireside chat on the emerging field of Gero physics, which I will introduce in a minute. We're going to have two experts, both who have been on the show before, Jan Gruber and Peter Fedichev. So we'll get to them in a few minutes. I want to remind you to use the Q&A function to ask questions, and we will try to get those questions to as many questions handled as possible. Before we do that, though, we have Belinda Wang, a research associate in Andrea Meyer's lab, and she will present on her own study, Healthy Longevity Conceptual Framework, or HELO.
Thank you, Professor, for the kind introduction. Hello, everyone. As mentioned, I'm Belinda, a research associate from the Academy for Healthy Longevity. And today, I'm excited to share with you our Healthy Longevity HELO conceptual framework. Before diving into HELO, let me provide the research background and why we explore this. Between 1950 and 2020, the global population tripled. As we can see on the graph, the number of people aged 70 years and above increased disproportionately. While people are living longer, with the global average life expectancy at 73 years, these additional years are not always lived in good health. Many older adults face chronic conditions like diabetes and cardiovascular diseases. This has created a gap of about 9 years between lifespan, the total number of years a person lives, and health span, the total number of years a person lives in good health. Healthy longevity medicine aims to bridge this gap by focusing on improving health span through innovative approaches. Using tools like artificial intelligence and biomarkers of aging, we can estimate a person's biological age and develop personalized strategies to prevent chronic diseases before they occur. Understanding public perceptions of this emerging field is essential to promoting healthy longevity within communities. Despite its potential, public understanding of healthy longevity medicine remains limited. A survey of 1,000 Americans revealed that 65% would live to 85 years with compromised health. When provided the scenario of guaranteed mental and physical health, 80% of them decide a lifespan of 120 years or more. This shows how the perception of ill health influences one's preference for own lifespan. Insights from the McKinsey Health Institute survey across 21 countries also highlighted that older adults value purpose, stress management, independence, and meaningful relationships as key contributors to overall health. These findings underscore the complexity of how people perceive health, aging, and well-being. That's why it's vital to explore public views to effectively design and implement strategies promoting healthy aging within the community. And this brings us to the aim of our research, to understand the awareness, knowledge, and motivations of the general population towards lifespan, healthspan, and healthy longevity medicine. To achieve this, we developed the Healthy Longevity Gero framework. This framework is based on a review of the literature, guided by expert discussions across disciplines. It incorporates evidence-based concepts related to health decision-making, aging, and longevity medicine. The Gero framework consists of three main components that interact and collectively shape one's views towards healthy longevity. The first one, awareness, which examines public familiarity with healthy longevity medicine and their impressions of the field. Second, knowledge, which explores public understanding of terms like lifespan, healthspan, healthy longevity medicine, and biological age. And third, motivations, which investigates the factors underlying one's motivations towards healthy longevity. These include personality, current behaviors, such as the use of health services, personal values and beliefs, such as religiosity, and health-related perceptions, such as ageism. Our next step is to design a survey using the Gero framework to explore public views globally. The Gero survey has been designed and is currently being implemented by the International Gero Consortium, which presently includes members from 8 countries. The survey has been administered in Singapore, Indonesia, Hungary, the UAE, and the Netherlands, with the rest to follow. The Gero survey has also been adapted to specific subpopulations, such as healthcare professionals or intergenerational families, to examine cross-cultural perspectives on healthy longevity. And that marks the end of my presentation. Thank you very much for watching.
Thank you, Belinda. We'll be interested to hear the results from this. From revolutionizing finance to inspiring breakthroughs in AI, theoretical physics has repeatedly transformed entire fields. Now it stands at the threshold of aging research, ready to decode the intricate systems of life. Tonight, a panel of leading experts will explore how physics-based models and computational techniques can uncover the fundamental mechanisms of aging, offering new pathways to extend lifespan. The discussion will also highlight the importance of robust quantification in modeling biological complexity, enabling precise predictions and actionable insights into the aging process. I should note that we're also going to have a conference on gerophysics, the inaugural conference here in Singapore in a couple months, and I will talk more about that at the end of the show. Tonight we have 2 panelists. Both of which have been on the show before. First, we have our very own Jan Gruber, who's an associate professor in the Department of Biochemistry here at the National University of Singapore School of Medicine. He obtained his PhD in molecular biophysics from Oxford University. He also has started and currently leading a C. elegans aging laboratory, which combining experimental models with computational biology to understand and intervene in the molecular mechanisms of aging, as well as age-dependent diseases. His research group focuses on studying biomarkers of aging, the role of mitochondrial and free radicals in aging and age-related diseases, and also aging evolution. More recently, he's been looking at a lot of human data and trying to do modeling to understand why we age and how to measure it. I'm going to pass on Jan for a second and come back to him in a few minutes, and we'll discuss his thoughts on gerophysics. Before we do that, however, I'm going to introduce Peter Fedichev, who's the CEO and co-founder of Gero, a data-driven longevity biotech company that develops new drugs against aging and other complex diseases using AI platforms. He obtained his PhD from the University of Amsterdam, and his scientific background lies at the intersection of physics and biology, with specific expertise in condensed matter physics, biophysics, and bioinformatics. Peter is an author of more than 100 published papers in multiple domain areas, including publications in journals such as Science and Nature Communications. His work has provided key contributions to the theoretical understanding of the aging process, the rate of aging, and the limits of lifespan and age reversion. I've asked Peter to give a short presentation tonight. We're not going to do the full-length presentation we usually do. We're going to keep it condensed so there's more room for a fireside chat. But he's had some interesting insights from his work recently, and I thought it might give us all a bit of a framework for what Gero physics looks like. Peter, thanks for joining the show, especially at 3 a.m.
Well, thank you very much for having me. Please confirm that you see the slides.
Yes, we're good.
Cool. So let me tell you in a moment what I think Gero physics is. But first, before I show you some results that we have obtained, I would like maybe to start with a philosophical comment. So in physics, there are different kind of levels of models and theories for complex phenomena, which I'm sure aging is no exception. So of course, everything is normally driven by experiments. So whenever people find some novel, interesting thing like superconductivity, I mean, first they give Nobel Prize to experimentalists who discover it. And then theoreticians and physicists normally work on creating what is called microscopic phenomenological theories. These are theories that do not actually explain what's going on, but they condense the experimental knowledge to a very few variables. So these variables are normally called microscopic or emergent variables. So once again, these things are not made in order to explain. These things are made in order to describe, to condense the experimental facts to a very few notions. And then all the other people are trying to build what is called microscopic theories that explain those microscopic models with the help of very few, with the help of all kinds of microscopic details. So what's important is that when you have this kind of microscopic description in terms of very few variables, is that you're able to capture all the diversity of the experimental information. And at best, you can arrive to the situation where you cannot, when you can avoid doing some of the experiments. You can check your microscopic theories against the experimental data before running the experiments, and that saves a lot of time. So I think what's going on now in aging, once we are getting lots of biomedical data, lots of biological data, molecular level data, is that we are kind of overwhelmed with the details. We know so much, we can measure so many things that we stop understanding what's going on. So what I think is happening right now in the field of aging is that we are trying to understand what is this kind of minimalistic description. Our aging clocks are a wonderful example of such condensed representation. And then if we can tie them together into a kind of coherent picture, we can probably start asking questions: what is possible, how aging works, what is possible therapeutically, and what we can expect in years to come in terms of practical life extension. So just to give you the ideas how this works, let me give you kind of a short overview of our latest results. So obviously, aging is a dynamic process in a very complex system, like an aging body. And of course, aging body does not only age, but also does a lot of useful functions. And from kind of very microscopic, or let's say thermodynamic point of view, what living organisms do, they transform energy into useful work, right? So they generate energy, activate molecular pathways, build their bodies. And of course, since in physics we know that no energy can be transformed to work with ultimate efficiency, they also generate heat, and that heat attacks the body, the molecules that the constituents of the organisms, in such a way that they produce damage. So on a molecular level, you can see it in such a way that if these boxes represent states of molecules, you could imagine that some of those states change together. That's how we activate pathways, perform biological functions. And as a result of these activations, we generate heat, and this heat attacks the same molecules in a kind of random stochastic way. And this stochastic damage may accumulate over time and cause the disruption in biological processes. So which means that if if you have lots of molecular level data, at some point you should have two kinds of processes, and I will show you in a moment that you always see them in biological data. So one process would be kind of a stochastic lifelong trajectory. This is simply the response of the body to all kinds of stochastic stresses and the green lights. These are the coherent features. And then there would be also another bunch of features that would just increase linearly on average and track the accumulated damage. You could argue, by the way, that this damage would be proportional to the metabolic output per cell. So this is exactly what you see in actual data. So on the right, I have examples from DNA methylation in humans. You could see two kinds of features. One of them in the bottom is exactly linear, and the other is hyperbolic, which I will not dwell too much into right now. The same you can see in mice. In mice DNA methylation data, you also have a cluster of features that is linear and the other cluster of features that is exponential. But what's most important that in any living organism, you can see a cluster of linear features. No matter how you cluster features, doesn't matter. Another important property of this cluster of linear features is that the features, the DNA methylation sites, or maybe other modifications of the molecules that contribute to this linearly developing cluster, they are not correlated with each other. So they are truly stochastic. One feature does not know about the other features. This is contrary to what you see in what is called pathways. In pathways, normally one thing is activated along with other things, and pathways are correlated excitations. So what we can see here is a hint that those features that are linear, that are tracking the damage, are possibly entropic, meaning that they are not correlated. There is no information in one against the state of the others, which means that if you act at one, you do not affect the others, which means that these features are very hard to revert. So they are probably microscopically irreversible. And that's, by the way, how physics comes into the question. So we start by describing what we see in the experiment, but then we start applying physical intuition to get the idea of what is changeable and what not. So just to give you the idea how this kind of different dynamic and stochastic entropic features respond to biomedical interventions, I will show you one example. This is parabiosis. Parabiosis is an intervention where old animals are connected to young animals. And normally old animals become younger and young animals become older. It has effect on lifespan. And when I see how these correlated features change in response to parabiosis, on the right, you can see that right after the parabiosis, the first columns, well, first the younger animals, yellow, are younger, this feature-wise, than the older animals. And then once the parabiosis happens, the older animals, the blue guys, become the rose ones. They become younger by these kind of correlated features. And the effect stays 2 months after the intervention. So these are the columns on the right. When I look at this linear cluster of this kind of uncorrelated stochastic features, you can see that younger animals are still younger, so they have less linear features, less stochastic features, but the parabiosis has no effect on these linear stochastic features, meaning that most probably these features are indeed entropic. Entropic means irreversible. So this kind of very simple, with not too much of an abstraction, just by observing features that we have in the data using single cell data in order to check cross correlations between those features, we can see that there are actually two kinds of biological clocks that exist in living systems. Some of them are dynamic, response to treatment, possibly reversible, and some of them are not dynamic, unfortunately irreversible entropic, meaning that they only kind of grow up in time, and this is the biological time in living systems. So this is actually a demonstration of the second law of thermodynamics. So with the limited time that I have, I will bring you another example how thermodynamics works in living systems. And for that, I will call for a very large data set, which was built by LTS Labs. And these guys produced DNA methylation of more than 100 mammals. And for those mammals, you can do lots of analysis. And what you can do is that you can look at all this data. You can try to cluster them at the same time. You can look at different clusters, relate them to biological function. I mean, that's what bioinformaticians do without any physics. But where the physics comes in is when you try to study the age-related features in this data set. So there are two age-related clusters. So on the left, they are plotted as a function of age, and on the right, you can see the plot for these two age-dependent features as the variance against the mean. You can see that there is one feature where the mean is linearly going up and the variance is linearly going up. So this is a hallmark of a stochastic noise. So all mammals, not only mice and humans, have this stochastic noise variable. And so which means that we can track this stochastic noise variable as a function of age. We can measure the rate of damage from the entropic damage from DNA methylation. And it happens exactly that the rate of this damage accumulation is inversely proportional to the development time, which we can also, which actually tells you that the rate of the damage accumulation is proportional to the metabolic output per unit mass. And this is, by the way, positive news. That means that aging, the damage accumulation, is a cellular level problem, right? Because this is metabolic output per unit of mass. Time is short here, so you can do lots of interesting stuff here. But if you look at the fluctuations of biological pathway activations, you can see that, like for atoms in the room, even though the living systems are incredibly complex, and I will have maybe 3 slides more, even though the living systems are incredibly complex, the fluctuations of pathway activations in different species are actually connected to each other. It's like for all molecules in your room, the temperature, there exists a parameter that is temperature that measures the fluctuations of the velocities of the molecules, even though they interact, collide, and everything else. In living systems, the pathway activation fluctuations, so the biological noise, is also controlled by a single variable across all the pathways. So this is another example of universality. So this effective temperature is not your body temperature, but nevertheless, it controls the biological noise across all different biological pathways. And then to cut the longer story short, I will tell you that this power of fluctuations, the effective temperature, in fact controls the difference between the mean and the maximum human lifespan. The maximum human lifespan occurs when damage destroys one of the leading pathways normally associated with innate immunity and kills the animal. So this is the maximum lifespan that you can achieve. And if there were no fluctuations, all animals would die at the same age. So the fluctuations that are controlled by this effective temperature actually controlling the difference between the mean and the average lifespan. And this is, by the way, a very large effect. So I will have just the last slide to summarize that. By building this phenomenology, right? So what I have just tried to guide you through, this is just an example how you take lots of molecular level features like DNA methylation and everything else and try to cast them in thermodynamics language. With this, you describe living systems with very small number of variables, and those variables have very specific meaning. Some of them are hinting at dynamics, so they tell you which of them are reversible and which are not. And some of them are emergent, like effective temperature, that are properties of biological noise, and they tell you about the character of mortality and lifespan. With this, we believe that you can do actually 3 types of drugs that will change human longevity. I mean, one drug that affects specific pathways, one type of drugs affect specific pathways. These are drugs against hallmarks of aging. The strongest effect will be the drugs against metabolic syndrome, and they will extend life by 10 years maybe. I mean, that's all you can get. Then there will be drugs that are controlling this new parameter that this is effective temperature. These drugs will bring average lifespan to the maximum lifespan. This is not 10, but 40 years of life. And then finally, there will be another kind of drugs that will change the maximum human lifespan. These are still a little bit futuristic, but what I've been trying to convince you that this kind of physics-based framework is a good way to classify work and actually guide the work towards the larger effects. This is the way how you can actually foresee. Remember, I started from that slide that this kind of phenomenology is a proven way to avoid doing most of the experiments, to focus your activity and energy on things that can have a larger effect. And I think this will be this eventually will lead to much better therapeutics that we're using now. Sorry for being longer.
Yeah, that's great, Peter. Well, you can go 3 minutes over if you get up at 3 a.m. Yeah. Maybe I'll come back with some questions in a minute, but let me bring Jan into the discussion first. So maybe, Jan, you can just tell us a little bit about how you're thinking about things physically and in terms of aging, and how that's guiding your research.
Yeah, okay. So fundamentally, I think physicists are trained to, from an early age, trained to take a really complex phenomenon and reduce it to the simplest possible explanation or a model that reproduces the major features of that phenomenon. And when I started out many years ago, I was trained as a physicist in biology. The problem was there wasn't a lot of data. Data was very low dimensional. It was very low n number. And so the only thing you could do is essentially ask hypothesis-driven questions and do hypothesis-driven experiments to try to sort of, build a model in your head over many years by exploring the phenomenon. And what has happened in the last few years or the last decade maybe is that we're generating hugely complex multidimensional data of complex phenomena at many layers of abstraction, at many layers of scale. And the problem has really flipped. Instead of having too little data to try to understand the phenomenon, we have arguably much more data than we can make effective use of. Not that data isn't still limiting, but oftentimes you can only explain one data set in its own context, and the attempt of, for instance, connecting it to some other type of data or another experiment from another group is almost impossibly difficult. And I think this is where physicists have something to offer by being used to applying things like symmetry and conservation laws, and aggressive abstraction to produce a model that produces the essence of the phenomenon. And I think that's where I'm excited seeing those two worlds come together. That's why I'm excited working with Peter. And I'm excited to see all the interesting single cell, longitudinal, multi-omics data coming out, because I think that then allows you to in turn test those models and those predictions using those data sets that are becoming available. And I think there's a real chance, and with the help with AI, which allows you to analyze data in different ways, to really make progress on problems which were essentially unsolvable with the manual, iterative, hypothesis-driven approach that biology has been using for the last hundreds of years.
Yeah, I think so too. I mean, it's amazing how fast data has changed the world. I mean, I have dry lab people in my lab doing bioinformatics. I never thought I would have so many of those people in my lab. It's like an invasion. It's good though. And I think that it's really going to turbocharge how we think about aging. I think that why physicists though? I mean, there are aging researchers that try to generate models too. What do you think is special about the world of theoretical physics that makes it so appealing to bring into aging research?
You're asking me?
Yeah.
Okay. I think physicists have a longer history maybe to get complex behaviors from simple causes. So like we can have relatively small models that produce really complex behavior, and we have tools to try to understand the phenomenology and the simple causes, and then to add additional complexity with perturbation. That's something physicists have a tool set for, and probably a head start in complexity science and dynamical systems. Whereas in biology, there's a tendency of essentially limiting the modeling to what you can do by drawing a cartoon, right? Oftentimes, I'm not saying biologists aren't using mathematical tools now, but I think traditionally there's a lot of difference in this approach. And so I give you an example, my own experience. We used to measure a lot of free radicals and oxidative damage. And one of the theories at the time was that we said, well, we have an exponential increase in mortality. And so we're expecting to see an exponential increase in damage in some molecule, or at least a nonlinear increase. So like an amplification, a feedback loop, a vicious cycle. And, I spent lots of years trying to measure damage, and you always ever get a small linear increase. It's statistically significant, but it's linear. And so, it took a while to realize that, essentially there's a very natural way of getting exponential mortality from linear change in parameters, from linear entropy, from linear damage. This is actually very natural. And if you write down the equations and you look at the physical models like Langevin equation, you can get that very readily. But many years we didn't really think about it this way. We essentially expected an exponential effect to need an exponential cause, and that I think is where physicists have far less, or so let's just say they have a head start in thinking in those terms.
Peter, you talked a lot about metabolic rate, and there have been theories that metabolic rate sort of defines aging going back almost to the point where theories of aging started. That a cell or an organism would have a limited capacity for metabolism, and that would lead to death. Are you going back to those models, or what? Because a lot of people sort of semi-rejected those models because you can find examples of animals with high metabolic rate that live a long time, or for instance, exercising a lot, which enhances metabolic rate, arguably extends at least median lifespan. So, how do you put these statements about metabolic rate in the context of those theories, and how do you put it all together?
Well, that's a great question, because I think there is a very strong evidence that this entropic damage is indeed metabolically driven. So I think that there is no known counterexample. The only problem is that you need another device, as Jan suggested, to relate this damage to actual lifespan. I mean, damage is damage. The damage does not necessarily even causes death. I mean, it's related to death, but you need to have something else. And lots of our intuition is unfortunately coming from shorter-lived species like mice, worms, and other guys, where this entropic damage does not have time on their lifespan. With their short lifespan, it doesn't have time to actually contribute a lot to the lifespan of these animals. These animals are dying for different reasons, so they are essentially dying because of this vicious feedback loop in one of their pathways. And of course, lots of drugs that act in that pathways, like inflammation-related drugs, extend life in those animals, and this happens totally unrelated to this entropic damage accumulation. So that's why I think here where the power of these models comes in, because you have to think, as Jan suggested, both on what is damage and how it is, and if it is related to lifespan. And you can find examples when it is not, or not so much.
Yeah, I think that's fascinating. One of the things you used to always, either of you can answer this, or you can argue amongst yourselves. That's fine with me. One of the things people used to always argue, because when I came into the field of biology, I think a little bit older than you guys. When I came into the field of biology, it was really non-quantitative, and when people started talking about systems biology 25, 30 years ago, which the idea was to try to quantitate, and things. And then, that was still a struggle. And so a lot of people were saying, the variables are just so complex compared to other fields of science in a living human being that it's never going to be possible to think about this mathematically in any effective way, at least not in the near future. It seems contradictory in a way, because now you can, talking about emergent variables, you seem to be saying that this very complex system boils down to two or three emergent variables, which I guess is an encouraging thing if you want to do something about it. Although maybe we'll come back to that point later. How does so much complexity distill itself down into simplicity?
Well, I think there is one field many people, talk. It's do not necessarily understand, but talk about quantum mechanics, or even about general.
Stop talking about me.
What I'm saying is that there is a public discourse about that there have been physics revolutions, like quantum mechanics, even relativity theory, that are discussed outside of physics. And then there are some revolutions in physics that happened, and Nobel Prizes were given in the 70s, especially related to renormalization group, emergence, where people understood how complex systems on large scales become, in a way, simple and universal. And these things did not, to my taste, penetrated public discourse, so they are not discussed very well outside of physics. And I think in many fields of science, even people still are very reductionistic. They think that by piling up data and writing equations and crunching numbers, they can go from the lower scale to the upper scale using the same terminology and approaches. So what this emergence is telling you that when interactions are high and systems are sufficiently complex, when you go up in scale, two things happen. First, new features emerge. That's called emergence, right? So new phenomena, new variables that do not exist on the microscopic scale. And the other thing, which is very relevant to medical sciences, I believe, is that the property that information doesn't flow up from the bottom scale to the upper scale, right? I mean, if a complex system is described by just a few variables, it means that all the other millions of variables that you can measure are not relevant. They're useless. Right? Information doesn't flow. So you do not see emergent variables on the microscopic scale, and that's where molecular biology likes to operate. And things that are relevant for longevity and chronic diseases operate on a medical scale. That's why we have different professions even, right? I mean, there is biology, there is medicine. These are sciences of different scales. So I think most of biological science still operates under the assumption that just with a little bit more effort, we will bridge precisely the bottom scale with the upper scale. And strong emergence tells you that this is not possible. So the right way to do is actually to define your phenomena on the upper scale, on the medical scale, and then try to go down and try to relate molecular level features that are associated with the microscopic scales. And I think as soon as this experience diffuses into biomedical sciences, we will have a lot more successes where people first get the right description on the right scale, on the medical scale, and then bridge. Don't study all the possible molecular biology. I mean, this is just random. Maybe if you give me just 30 seconds, I like to bring this example. When people devised steam engines, they didn't know about molecules. And we were lucky because assume that by that time somebody would bring up a device that would measure positions and coordinates of all atoms in the steam engine. People would be writing, publishing Nature papers about 3 atoms moving right together with the piston at the same time, right? So most of the information about positions and velocities of atoms, even if they were measurable, are useless. So I think that's what you're precisely referring to in the biological sciences. Now we're in the situation where such a device exists. We can measure millions of features, but they're still useless. That's
I guess, though, do you need it? I guess it doesn't matter what features you measure, pretty much, but you needed some measurements of millions of features to transform the data into your observation, right?
Statistical, I mean, what this science about emergence is telling you that only statistical properties of those millions of features are important, not any specific features, right? So that's where the kind of flip in the understanding comes. Only statistical properties are useful.
So go
ahead, Jan, please.
So you were just saying, like, you're a bit older than us only, but I do remember the Human Genome Project, and I also remember the first qPCR machine appearing in the lab. And so, it went from, you don't really have a genome for most animals you work with. You have to map genes very carefully and by hand. My wife spent a lot of time mapping a single rat gene in the lab. And you would run gels like ballistic PCR or something to look at gene expression of a single gene. And this forces you to an extreme degree of reductionism because every single time point is a week of work. And then qPCR came about and we all thought, okay, this will break open everything. We can do tens of genes, right? At different time points. And then microarray and then, RNA-seq and so on and so forth, next generation sequencing. And so we have essentially many orders of magnitude more resolution in data now than we had when I was a student. And at the same time, computers have become millions of times more powerful. And the first thing that happened when this data came about, when this early systems biology happened, is that people wrote very large differential equation systems where almost all the parameters were guessed and like unknown by orders of magnitude. And those systems almost never behave even approximately like the biological phenomenon because you can't get it from the microscopic description. And a lot of time the mantra is bigger computers, more equations, more data points, more data. Eventually the model will behave like the real thing. And I think the truth is, to use Peter's analogy, is like what we really want is the thermodynamics of the phenomenon we're interested, right? We want to figure out what's the pressure, the temperature, of the piston, and what are the 4 or 5 variables that describe it can be abstracted. And yes, it might need the high dimensional data. And yes, the high dimensional data, multi-scale data allows us to test these hypotheses. But I think having a thermodynamics of the processes that we're interested in is illuminating. And I also think it's interesting that in medicine, still most discoveries aren't made by like microscopic description of the disease process, but rather, phenomenologically or serendipitously discovered interventions that then often end up working in diseases completely different from the original discovery. And that's because, as Peter says, medics operate at a much higher level, trying to perturb the system and watching what overall happens. And so I think having the physics in between those two extremes is very interesting. It's very exciting that that's now possible.
I'm gonna we'll come to questions in a minute, but I'm going to, before we do that, just raise this issue. So, we had you mentioned three classes of drugs. Class 1 drugs, which are what Jan and I have been working on mostly, which are going to have significant and important effects, but not large effects on a grand scale of aging, maybe 5 or 10 years. In humans, yeah. And a second class of drugs, which will, maybe affect that some of those thermodynamic properties and reduce the noise in the system and have a bigger effect. And then you had a third class of drugs, which is much less well defined, where a survival curve is not going down anymore with time, at least because of aging. So maybe you could, I think we all know what the class 1 drugs might be. We've talked about them on about 100 different shows. Maybe you could comment a little bit, to the extent you can, about what a class 2 drug looks like, and then speculate as to what a class 3 drug might look like.
Well, first of all, I'd like to comment that this phenomenology is kind of generic, but in different species, unfortunately, we can have different molecular mechanisms that produce the best drugs of each kind. I mean, this is the famous translational problem. I wanted to put the note on it. So yes, you correctly explained that we believe that in long-lived species like humans, individual hallmarks of aging are kind of controlled most of the time. We don't have any exponentials, as Jan said, on hallmarks of aging in humans, and therefore we believe that biological noise has a lot more effect on the lifespan than the dynamics of individual hallmarks of aging. So the reasons for biological noise could be different, but just to hint is that obviously you have more noise where you have fewer molecules. I remember you corrected me once when I said that there is one DNA in the cell. There are a few more, but still, most of the molecules are more abundant than DNA. DNA is very it's packed in a very complex way, and lots of proteins participate in packing and binding, initiation of all kinds of processes, and even small discrepancies between on and off rates on those proteins interacting with DNA could generate a lot of noise. So we believe that more focus should be on proteins that directly interact with DNA, and as the major source in the cell. And the challenge is to find for every particular species, to find the most vulnerable set of targets that is associated with the noise.
Are we ever going to get to reversing aging if it's entropy?
Well, my point is that never say never, right? Because the amount of complexity that you can reduce obviously scales with computation, AI, and everything else. But what is nice about doing prediction in medical sciences is that since development and clinical trials takes 10 years, you can safely predict what will happen in 10 years from now, right? I mean, to get something working in 10 years from now, you have to have something working already, at least in the lab at this particular moment. So if you let me to make the statement like, would it be possible within the next 15 years, my guess would be that age reversal is mostly impossible. Small effect will be achieved, but not the large effect. But I think aging can be entirely stopped with this kind of science within 15 years or less.
Jan, you want to comment?
Comment on?
That question, yeah. when are we gonna? I want to dunk a basketball again. When am I going to be able to dunk a basketball again?
So the joke is always it's really hard to make predictions, especially about the future. But fundamentally, so let me just evade the question by saying I'm really excited about the effective temperature as a target in the meantime, while the other ideas are getting explored, and about dissecting the phenomena. So one of the things I think that is hugely valuable in this kind of work is that it tells you what signatures you're looking at. So, for instance, it's always a problem when we build clocks to distinguish a disease signature from an intrinsic aging signature, an entropic aging signature. And knowing what you're looking for, having a model that says you'd expect those two phenomena to happen and they overlay in this way allows you to build the right kind of tools to distinguish those two things. And I think that's something we absolutely need if we want to slow down or stop aging, because you need to get at the underlying cause, not at the coupled disease process, although that's useful, but that's always going to swamp the data, right? An exponential process is always going to be the biggest signal if you just naively look at what changes over time. And I think that's why I'm extremely excited about this. Whether or not reversal is possible using, reprogramming or regrowing tissues outside of the body and building new organs, or, as Peter says, of order one transplantation. I think those are questions that are probably not going to be answered in the next 5 to 10 years. That's for sure. But the other ones maybe.
So I could keep talking, but I think instead we'll bring in Trina Tan, who's a master's student in NUS School of Medicine. She's got a lot of questions to choose from, so choose wisely.
Thank you. Okay, so we have a few questions. So the first question is, what distinguishes Gero physics from other interdisciplinary fields, and why is it crucial to focus on this area at this time?
Well, I would answer with a joke. The Professor Rutherford, the guy who got the Nobel Prize for discovering the inner structure of the atom, joked once that there are only two kinds of sciences, physics and stamp collection. So I think that by bringing physics into biology, we move up in the food chain of sciences in this way.
So I would say at the beginning of most or many aging papers, it says aging is a complex multidimensional process that, is difficult to detangle or describe or analyze. And I think that means that, having a perspective from physics there is useful in the conversation. I'm not saying that that's the only way you should look at, but I think it's welcome and useful to have that input. And I think it is now the right time to do this, just because we're generating the kind of data that we're generating. As a side note of this, and I'm going to shut up in a second, but we're still producing orders and orders of magnitude less data than a lot of physics experiments, right? If you do an experiment in the lab that you can just rerun and get millions of time points on, that's still something in biology we can't do. But we can now generate, tens of thousands of points, and that's makes those sort of tools actually applicable. So I think it's the right time to do this, and it's a complex phenomenon, as we all agree. So it's a useful place to apply those tools. And it's the one I care about, of course.
Okay, thank you for your insight. Then we have a next question. Can you elaborate on the key scientific principles that underpin Gero physics and how they integrate geroscience with physics?
Do you want to go?
Yeah, maybe. I'll say it's geophysics is very young. I think University of Singapore is just producing the first conference on that, so it's maybe too early to say about generic principles of that. But I think overall the principles are shared with other applications of physics to complex systems. It's emergence, maybe strong emergence, like inability to go up with the information about microscopic details, up the idea of building microscopic model, identifying effective microscopic variables, emergent variables, very few of them, focusing on the very few that contribute most to the phenomena of your interest. I mean, there is the whole tool set. There are books how to work with novel complex phenomena like aging. So I think we are on the brink of discovering this aging phenomenology, the minimum set of the minimalistic models that to my taste, we need to find models that describe both aging and non-aging mammals as the first immediate goal. And once we will agree on those models, we will understand which phenotypes are actually responsible for dramatic life extension, and we will focus our energy on drugs that will have large effects.
Yeah, and if you, again, to reiterate the book point, there are books on emergence and complex system and dynamical systems. You can pick them up and flip through them, and they have examples from biology, and there are things like circadian rhythms and, genetic switches. There are examples explaining the stock market or aspect of the stock market, the weather, and so on and so forth. But there aren't chapters on aging in those books, by and large. I looked. And so there should be. And I think that's where the geroscience comes in. There should be a chapter on, application of complex system science and emergence to aging phenomena, because that's an important problem to apply it to.
If you let me also comment, like some of the people who were giving lectures to me in physics were saying that if you have a new complex phenomena, you apply a textbook to it. So in the worst case scenario, you will just describe this thing in a useful way, or you will do a revolution in physics, which is much less probable. So I think it's very important to use the knowledge and experience that was gained with a lot of other complex systems before, like stock exchange, weather, and everything else. And there is this wonderful thing in physics which is called universality. There is this surprising ability of kind of the same models describe different phenomena, and I think people should use the experience from other fields of other successful applications of modeling, because I think it saves time and gives lots of intuition.
Okay, so we have one question. What are the biggest challenges in applying physics-based models to complex biological systems like aging?
So I think, from my perspective, one of the biggest challenges is still the sparsity of data. I know I'm contradicting myself in a way, but there are really cool questions we could ask. For instance, if we had much higher dimensional longitudinal data in many people and many species. And so that's why, for instance, the recent preprint takes advantage of a comparative biology data set of epigenetics in many animals. And that is a giant data set, very expensive, very impressive. But of course, when you start playing with it, you wish it was 10 times larger, right? And I think this is still true, that biological data is extremely expensive and time-consuming to generate. And one of the challenges is to get data that sufficiently constrains the models that you can rule out or prove what you've just derived based on what the data can actually support, and that's still difficult.
We were talking about the quantity of data. What about the quality of data? Is it sufficient in biology to model?
So I remember, so the answer is, it's difficult, right? Many parameters in physics, to many digits after the decimal point. In biology, I remember we did modeling on some mitochondrial things in the past. And the simple question that the people I worked with who were modelers asked is, what is the amount of mitochondrial DNA damage? How many damaging lesions per base pair are there? And we went to the literature, and we got initially a range which a factor of 10,000 between the highest and the lowest value. So we didn't know this to one digit after the dot. We knew it to a factor of 10,000. So when you're trying to constrain things, with that kind of uncertainty, it's incredibly difficult. I think the world is better now, but I still think it's much harder to measure something to many digits of precision.
Yeah, I would comment here is that it's not only kind of molecular level data that is not known. Even if you try to, for example, compare the predictions of your models for average lifespan for mammals, you will find that there is a ridiculously small amount of data, even for such kind of important things like what is the average lifespan of different species in different conditions. So I think the problem with the data exists. The problem with the quality of the data exists. I think most of the data is collected in shallow experiments where people produced a few hundred samples for a particular experiment with a very particular design of the experiment in mind, which prevents this data from being used across different experiments in kind of meta-analysis way. But I think think this changes very quickly. So the amount of biomedical data is going up, especially for humans. So I'm very optimistic here. I think the other problem is maybe still kind of cultural. Lots of people describe complex phenomena, and I think more attention should be paid to explaining complex phenomena. It's better to have a bad theory than no theory at all, even if you have a very complex data set. So I think it's partly technical, but also partly cultural.
Yeah, sorry, one last thing is the technology moves so fast, right? So even if you want to look at gene expression, it's very difficult to compare microarray to like early transcriptomics, RNA-seq to the latest data sets, and now it's single cell. So it's actually almost a digital archaeology involved in trying to figure out how the data was generated if you want to aggregate, the past record. And I think that's also a challenge that needs to be addressed.
Okay. There's one question which asks, what educational or training initiatives are necessary to cultivate expertise in geophysics among upcoming scientists?
Well, I mean, cross and interdisciplinary training, right? Having biologists learn about linear algebra, it would be useful. And, actually a lot of this is happening in the guise of machine learning, right? Like when we learned linear algebra, it was completely abstract. It was a super important first math class, but there would never have been something a biologist would have been found to take at my time. Nowadays, people do that because they want to learn about machine learning and they learn those same mathematical tools. So I think that's a great thing. And vice versa, having, physicists in the lab and having them actually experience how maybe some of the data is really hard to generate and you appreciate what it takes to do the data set would be useful. So I think exchanging across the silo is always of value. And I think a lot of this is happening now. I remember when, the only math class biologists would take would be a statistics class, basically. And it stopped essentially at, t-tests and maybe ANOVA. That was the sum total. Very different with the students today. There are many more people who cross the boundaries.
I'm one of the only biologists that got a math major, and the only class I didn't take was statistics. I have no idea what I was thinking.
There is an interesting question. It says, explain geophysics to a 10-year-old and bring an example from your own life.
Oh,
10-year-old. Well, maybe applying things we know that are true about the universe to biology. Like, deep things we know and applying it to biological questions. And an example is, oh God, what is an example? I have 10-year-olds, so I should be able to do this. What is an example? Trying to build...
Yeah, my 10 years old has infinite trust in his parents and believes that aging will be solved before he gets out of school.
Mine.
Give him a couple of years.
Yeah, so probably we'll skip on that. Other than, look, if aging is indeed entropic, time is a little bit more valuable. So maybe that's the only thing that we need to explain as a practical application of geroscience to a 10-year-old. Time in school is valuable.
Yeah. What was the quote? Physics is the law, everything else is a suggestion, including evolution, who has to find a solution within physics.
Which has to work
the mindset.
Yeah. To your previous question, I didn't comment, but now I probably got an interesting comment. So when I was not 10 years old, but a little bit older, I was thinking about between biology and physics. And, elder guys, wise guys told me that young boy, go to physics because there are cases when physicists go to biology, but there are no cases when biologists are going to physics. So now I know a counterexample. There is Stuart Kauffman, a great theoretician, the guy who started in neurobiology and turned into physics. And there are lots of good books. You can search for him, Stuart Kauffman, popular books on Amazon, everywhere. And this guy actually, because physicists are arrogant, as you can see from me, and that guy is the only known to me physicist coming from biology to biology. And he writes how physics is different from biology. So I would highly recommend, if not taking physics classes, but at least try to extend your intuition by reading first maybe popular books by people who think across these two disciplines, and maybe eventually taking classes.
Okay. Is there any time for more?
No, I mean, I don't think that I'm that old to teach young people, so I still hope to do some interesting stuff.
Maybe one more question. Pick a good one, and we'll end it on that.
Okay, last question then. Which is more important, the biology of aging or the physics of aging in longevity research?
Well, I believe in convergence, that things that work are not distinguishable from each other. So I do believe that biology and medicine is struggling a little bit with aging. Aging is the best phenomena to be described by physics and the whole biomedical sciences, to my taste. And I think that, we still don't have a solution against aging, which means that other approaches are required. I hope that this is a combination of machine learning and physics. Maybe it's just pure AI, AGI, I don't know. But why should we surrender so quickly? Let's try to apply physics first.
It's not an either or thing. It's like, that's the whole point about the Gero physics. It's trying to apply some of the tools from one domain to another. And that is often very fruitful, right? If you look to the history of science, for instance, when crystallography came about, there's a lot of application of physicists moving into biology and trying to explain how protein structures work. And, the central dogma, and it was extremely fruitful, leads to Nobel Prizes. And that's often the case when you move tools from one domain to another. In fact, if you look through the history of science, it's really interesting. There are often the same tools under 5 different names who get to revolutionize different fields simply because somebody learns something from one domain, brings it over, gives it a new name, applies it to a new problem, and everybody's very excited. And I think that's just how this works, right? You apply tools that you know from here, apply them to a new problem, and you find new solutions. And that's all that means. Yeah. It's not either or. You're not abandoning biology because you do physics.
Exactly.
So, great. Go ahead, Peter. Last comment.
Yeah. So we started with this idea of emergence and strong emergence. It means that physics will never eat all the other sciences, right? That would contradict the idea of emergence. So to my taste, physics is the only science at this time recognizing emergence. And that's why it's somewhat useful on the other levels out of its particular domain expertise. But that's it. It's not like physics is eating everything else for breakfast. It doesn't. There are other sciences as well, but I hope that physics view is useful, but it's not like definitive.
Great. Thanks a lot, Peter. You can go back to bed. And thanks, Jan. We've had you both on more than once, so expect to come back sometime in the near future. Thanks, Trina. Great job choosing the questions. I have a lot to get through, so don't leave. We're going to check who leaves, and you're going to get angry letters from me. Use the chat function, and panelists and all attendees options to leave comments and feedbacks on the show. Particularly tonight, it's useful because we had a panel instead of one single speaker, which is what we usually do. That creates a lot more conversation. I like it. Let me know if you like it, and we'll try to do that more often. We have several things coming up. The Healthy Longevity Intensive Course 2025 for healthcare professionals will be on February 25th and 26th of this year, followed very closely by the Founders Longevity Forum Singapore, Unlock Healthy Longevity. This event's happening on the 27th and 28th of February, and the idea is to bring together investors, innovators, entrepreneurs, researchers in biotech, clinicians, and in consumer longevity. And I'll add that physicists can come if they want. Register your interest now. Maybe more relevant for physicists, though, is the inaugural Global Conference on Gero Physics. That will be on March 5th and 6th of this year, and you can learn how physics can advance longevity research and practice. We're calling for abstracts now. The registration for the public will be open this weekend, so stay tuned. Finally, we have a video I want to point out to you. If you have Netflix, there's a recent video called Don't Die. Andrea Meyer and I were both involved in this documentary, so you can see us on it if you want. It tells the journey of the fight against unhealthy aging by the billionaire Brian Johnson. I don't think Andrea and I, or either of us, are endorsing exactly what he does, but it's an interesting show to watch because you can kind of see a hacker's approach to solving the aging problem. Andre and I shared a lot of input there, but there were a lot of other international experts you can listen to as well. Our next show will be February 6th. We're going to skip next week. And that will be Kenneth Meyer, a professor and group leader of the Human Movement Sciences Research Group at Maastricht University. The final video I'm leaving with is the #1 antidote to aging according to Big Think. Thanks a lot for joining us.
So usually when we think of changes associated with old age, we think of functional changes. Things like how fast you can run or walk.
Physical activity is important because when you're physically active, you stress your body. You produce what's called reactive oxygen species, which cause cellular damage throughout your body. It causes mutations, it causes inflammation. But because that's normal, our bodies turn on all kinds of repair and maintenance mechanisms that counter those. And as we get older, that repair and maintenance becomes really important because it prevents senescence. It prevents our bodies from decaying.
Stress is designed to make us stronger, whether it's emotionally or physically. Somebody has osteoporosis or osteopenia, right? Their bone density is becoming compromised with old age. Okay, well, why do we recommend working out, in particular with weights? The idea is that stress is an injury. We're injuring the muscles, we're injuring the bones, right? The bone remodels and becomes more dense.
Your bones are an endocrine gland. They release a hormone called osteocalcin, and it enhances the memory storage in young people, but also enhances memory storage in old people. And one of the reasons that exercise is important is because exercise builds up bone mass. This is particularly important in women, where bone mass tends to decrease more dramatically than in men, but it's important for everybody. So when you exercise, you increase your bone mass, you increase osteocalcin, and you improve age-related memory loss.
The evidence is incontrovertible. The more we age, the more physical activity is really beneficial.
So everybody wonders about what is the best time to work out. And my first answer to that question is always anytime you can fit it in from morning till night, fit it in. Physical activity is the most transformative thing that you can do, not only for your body, but for your brain as well.
The important thing is to get, be physically active because some is better than none. There's no magical number.
I like to set smaller goals for myself, doable goals. That is the key. Maybe it is a walk on the shore, a walk to the gym, a walk around the museum with friends, gardening. People think those things don't count. They all count. Be more broad in your definition of bringing more movement into your life.
What we really want to do is keep people healthy and functioning for as long as possible. And if that results in a longer life expectancy, that ends up just being the added bonus.